All right, Ninja Nerds, in this video we are going to talk about the auditory pathway. So if you remember, we said that there was two parts of the vestibulocochlear nerve, or cranial nerve 8. There's the auditory branch, or specifically we should actually call it the cochlear branch of the vestibulocochlear nerve, and then there's the vestibular branch of the vestibulocochlear nerve. In this video, we're going to talk specifically about the actual cochlear branch of the vestibulocochlear nerve. All the destinations, all the different types of circuitry that it's going through, from the inner ear all the way up to the primary auditory cortex. All right, so let's go ahead and get started. So if you guys haven't already, please go watch our video on the cochlea, where we talk about the actual spiral organ of Corti, because I'm not going to go into the entire mechanism, okay? But what we're going to do is we're going to take the scenario of hearing something, right? So I'm speaking, right? So when I'm speaking, I'm producing sound waves, right? Those sound waves, what happens with them? Well, if you remember, we said it starts with the auricle, or the pinna, right? So it's your ear, right? The outer ear. What does it do? It funnels the sound waves into a nice little tube. What is this tube called? The external acoustic meatus. From there, it hits a connective tissue structure called the tympanic membrane. The tympanic membrane will then vibrate, right? It can compress and decompress. When it does that, it shakes some little bones inside of the ear, right? And these bones in order is going to be malleus, right? Then you have incus, and then you have the next one, which is actually going to be called stapes. And what does these guys do? So once you actually vibrate the tympanic membrane, it vibrates the little ossicles, malleus, incus, stapes. Stapes starts tapping on the oval window. And if you remember, the oval window was connected to that nice cochlear structure, okay, which caused the fluid-filled vibrations. So now, if we come over here, let's pretend that this part right here is the oval window, and this one down here is the round window. And who was tapping here? Stapes. Stapes was causing fluid-filled vibrations through the scala vestibuli. And then what happens is, depending upon the frequency or the amplitude of the actual sound waves, it can cause vibrations within this basilar membrane, which is a part of the scala media. Also, you can call it the cochlear duct. But we said if the frequencies is not going to be high enough, right, then it can go past the actual cochlear duct at the helicotrema and then come down through the scala tympani, and it can push on the round window, right? So we said that. But let's assume that the sound frequency and the amplitude is enough to stimulate the actual basilar membrane and cause it to vibrate. If it vibrates, then what happens? We said that when the basilar membrane vibrates, it causes these little hair cells to move, right? And these little hair cells, they have specialized structures on their apical membrane. What is those called? They're called stereocilia. We said if the stereocilia beat towards the kinocilium due to them shearing across this red membrane, which is called the tectorial membrane, right? So if the actual stereocilia beat towards the big one called the kinocilium, it'll cause them to depolarize. If it depolarizes, it'll stimulate the release of a neurotransmitter onto these afferent nerve endings, which is called glutamate. Okay, so now, if you remember, we talked about this in the video on the cochlea. There was a ganglion, right? There was this ganglion right here. This is that pseudo-unipolar neuron, right? Has a cell body with two processes. One process is going out to the hair cells. They call it the peripheral process. The other one is going into the central nervous system. That's the central process. What is the name of this ganglion, if you remember? It was called the spiral ganglion. And that should make sense because it's going to innervate the spiral ganglion, okay? The spiral organ of Corti, all right? So it's the spiral ganglion is innervating the actual spiral organ of Corti. Now, peripheral processes go out to that organ of Corti, where the hair cells are. Central processes go into the central nervous system. Now, I'm just drawing one of these actual ganglion, but imagine for a second that I have multiple of them. So let's say here I have, again, here's a peripheral process, here's a central process from this ganglion. Let's say I have another one here, and here's a peripheral process, here's a central process. I'm going to have hundreds and hundreds of these guys coming together. And whenever they come together, they're going to make a specific branch. They call this branch here the cochlear branch of the vestibulocochlear nerve. So this is the cochlear branch of cranial nerve 8, which is the vestibulocochlear nerve. Because there's two branches, you know? The cochlear branch, which is coming from the cochlea, picking up sound waves, and the vestibular branch, which is coming from the actual vestibule and the semicircular canals, picking up static and dynamic equilibrium. Now, this cochlear branch of the vestibulocochlear nerve, he's going to start moving. We're only going to focus on the cochlear branch in this video, right? So what happens is this guy starts moving towards the actual pons medulla junction. But in order for him to be able to get there, he has to go through a hole within the skull. So there's a hole within the skull, specifically right around the actual temporal region, okay, the actual temporal region, or the petrous part of the temporal bone. There's this hole in the skull, and they call this hole the internal acoustic meatus or canal. Now, there's another nerve that actually runs through there. If you want to know, there's actually the 7th cranial nerve. So there's another nerve who actually does run within this hole here, and this nerve is actually called the facial nerve, cranial nerve 7. Okay? So there's another nerve that actually runs within the internal acoustic meatus. So now, what have we gotten to? First point here, we have the stimulation of the hair cells. Second point, we activate the peripheral processes of the spiral ganglion. Third part, we're activating the actual central processes of the spiral ganglion. Eventually, all these axons of the spiral ganglion come together and form the cochlear branch of the vestibulocochlear nerve. It then runs through the petrous part of the temporal bone, specifically through a hole there called the internal acoustic meatus, with another nerve going with it. All right, and this is going to be the facial nerve. Once it actually reaches this pons medulla junction here, it comes to specific nuclei. Okay, it comes to specific nuclei located within the pons medulla junction, specifically in the upper medulla. All right, there's a whole bunch of nuclei here. This nuclei as a group, let's pretend that I kind of circle all of these nuclei here. I'm going to circle all of these nuclei. These nuclei are called my cochlear nuclei. Okay? But what happens is we actually separate it into two parts. Okay? So these blue ones here, these dark blue ones, this is going to be the anterior or ventral cochlear nucleus. Okay? So this ones up here are going to be the anterior or the ventral cochlear nuclei. Okay? The one in the back is going to be the dorsal or the posterior cochlear nuclei. So this one right here is going to be the dorsal cochlear nuclei. Okay? So we have two parts here. Once these fibers come in, this cochlear branch of the vestibulocochlear nerve, it fans out onto multiple cochlear nuclei within the actual pons medulla junction. The nuclei that are situated anterior are called the ventrocochlear nuclei. The ones that are situated posterior are called the dorsocochlear nuclei. Now, technically, another thing happens. We technically separate the ventrocochlear nuclei into two parts. This is where it gets a little funky here. So we separate these guys into two parts. One part is going to be a little bit more posterior, and one part's going to be a little bit more anterior. So they call this one right here the ventral-posterior cochlear nucleus, and they call this one the anterior-ventral cochlear nucleus. All right? So it's a little odd, but yeah, that's what they do. Okay? So the ventrocochlear nuclei is actually split up into two parts, the ventral-posterior cochlear nucleus and the anterior-ventral cochlear nucleus. There's a reason why they actually kind of separate this for specific types of cells that make them up. All right, and we'll briefly mention that. Okay, so now, once these actual cochlear fibers, boom, synapse on these suckers right here, a lot of different things are going to happen, so just stay with me, okay? Let's start with the dorsal cochlear nucleus. The dorsocochlear nucleus has a bunch of different cells, but the main ones here in the dorsocochlear nucleus is going to be of two cell types. One is going to be called principal cells, and another one is you're going to have what's called stellate cells. Okay? What happens is the stellate cells and the principal cells give off their axons. And these axons, what they do is they start moving over, and they go to a special structure here located within the pons. It's called the nucleus of the lateral lemniscus. We'll talk about that in a second. But if you look here, the principal cells and the stellate cells, they give off these axons, okay? And as these axons cross over to the other side, they're also ascending at the same time. We call this fibers, or this tract, if you will, they call it the dorsal acoustic striae, okay? So they call these fibers here the dorsal acoustic striae, all right? They cross over to the contralateral side, and they pretty much synapse on a special nucleus within the pons, which is called the nucleus of the lateral lemniscus. So what is this nucleus here called? It's called the nucleus of the lateral lemniscus. Okay. Then we go to this next one. The ventrocochlear nuclei branch into two parts, the ventroposterior and the anterior ventrocochlear nucleus. The ventroposterior cochlear nucleus is mainly made up of cells. I don't know why I laugh every time I think of these guys, but they're made up of what's called octopus cells, okay? It's a real thing, I know, but they're called octopus cells. Okay, so the octopus cells are going to be specifically situated in the ventroposterior cochlear nucleus. What happens is these octopus cells give off their axons, okay? And these axons, what they do is they do the same thing. They cross over. They don't ascend as fast, but they cross over, and then they actually move up. So they cross over to the contralateral side, and then they move up. Some of the fibers might actually synapse on the nucleus of the lateral lemniscus. Some of them might not, okay? Most of the fibers that synapse on the nucleus of the lateral lemniscus is the dorsal acoustic striae. But now, these octopus cells, they give off their axons from the ventroposterior cochlear nucleus. The axons cross over to the contralateral side. Very little of them will synapse on the nucleus of the lateral lemniscus. Most of them will continue to move upwards, okay? What is this structure here called? Well, this is the dorsal acoustic striae. This one, I bet you, is going to be the ventral acoustic striae. They also call it the trapezoid body. So this one's got to be in between. So they call this the intermediate acoustic striae. Okay, so we have the intermediate acoustic striae, which is formed by the axons of the octopus cells. Then we have another one, the last one, the anteroventral cochlear nucleus. This one is going to have a bunch of different types of cells here. Some of these cells here, you're going to have the actual stellate cells again. So this is going to have some of those stellate cells again. And you're going to have another one. And these are called bushy cells. And there's different types. The main ones are going to be what's called spherical and globular bushy cells. Okay? Now, what happens here? When these guys are stimulated, they give their axons, which also are going to cross over to the contralateral side, some of them. Here's what's different about this one. When it crosses over, most of the fibers here synapse on a specific point. They synapse on these nuclei right here, okay? So they synapse on these nuclei right here. Some of them go to the contralateral side, and some of them also go to the ipsilateral side. Okay. When they do this, okay, what is this structure here first off? Formed by the axons of the spherical, globular, bushy cells and the stellate cells. This one has two names. You can call it the ventral acoustic striae, or you can also, sometimes you also hear it as what's called the trapezoid body. Okay, sometimes you hear it as the trapezoid body. Now, The ventral acoustic striae crosses over. Most of the fibers cross over to this green structure here within the actual lower pons. Some of them will stay on the ipsilateral structure within the lower pons. What is this structure here called? This structure here is called the superior olivary nucleus. So they call this the superior olivary nucleus. This structure is super, super cool, all right? There's two parts of it. We'll discuss it in a second here. Okay, but again, these fibers, most of them will cross over to the contralateral superior olivary nucleus. Some of them will stay ipsilateral. When they cross over, they call that the trapezoid body more specifically. But again, you can actually interchange that with the ventral acoustic striae. From here, from the superior olivary nucleus, these fibers can go up. So now you're going to have these fibers coming up here and moving with the dorsal acoustic striae, moving with the intermediate acoustic striae. And what is going to happen? Even some of the fibers from this, what is this structure here? So some of these fibers might actually continue here with the ventral acoustic striae, the dorsal acoustic striae. What else are you going to have? You're going to have the fibers from the nucleus of the lateral lemniscus. So this nucleus of the lateral lemniscus, some of these dorsal acoustic striae fibers are going to synapse there. Then what happens? They move up also. Look what you got here now. So now you have the dorsal acoustic striae, the actual ventral acoustic striae, which synapse on the contralateral superior olivary nucleus, and these intermediate acoustic striae. As they're moving up, some of them synapse on the nucleus of the lateral lemniscus. The more prominent one, though, who does that is going to be this guy here, the dorsal acoustic striae. Some of the fibers might not synapse there, though. Then you get this actual nucleus here. He can give off axons that actually move upwards. Now, as they're moving upwards, they form a tract, if you will. Okay? How would you define a tract again? A tract is defined as a bundle of axons within the central nervous system. Well, this tract is more flattened. And so because of that, it takes on a specific name, which is lemniscus. They call it like a kind of a flat tract. What is this whole tract here called? It's called it's ironic, the lateral lemniscus. So this whole tract here, this whole bundle right here is called the lateral lemniscus. Okay? Now, as these guys go up, there's some interesting things that can happen. There can be some crosstalk. There actually can be some crosstalk between the actual nucleus of the lateral lemniscus from both sides. That's really interesting. Because hearing is not just purely basically ipsilateral. It's contralateral, bilateral. Okay, so if we actually have any damage to these structures here, we still might be able to hear. Okay, on both sides. It just might be a little bit weak on one side. Why? Because the fibers coming from the other ear are also going to cross. So hearing is bilateral, which is really, really interesting. Okay, so again, nucleus of the lateral lemniscus can have some crosstalk. Now, where does this lateral lemniscus structure go? It primarily synapses on this structure located within the midbrain. What is this structure located within the midbrain called? Okay, this is a really important structure. Almost all of these fibers usually should pretty much all synapse on this structure. They call this structure the inferior colliculus. Okay, so they call it the inferior colliculus. So what is this purple nucleus here called? They call this the inferior colliculus. Colliculus, whatever. Okay. So now, from here, the inferior colliculus is really important because the inferior colliculus is actually, there's another structure right above it called the superior colliculus. You know, whenever someone yells at you or says something, you know, sometimes you turn your head that way. You focus your gaze on whatever someone is, where the voice came from. That can happen. The inferior colliculus actually controls a tract that goes downwards, a descending tract that goes to muscles that can move your head and your neck. So where you can fix your gaze on wherever that auditory stimulus came from. That's called the actual auditory reflexes, right? So this can actually become a part of a tract that can go downwards And this tract that can actually go downwards, we'll talk about this when we get to the descending tracts, but this can actually form a tract called the tectospinal tract. And this plays a role with your auditory reflexes. Okay, so if you hear something and you move your head that way, or your neck muscles, your head muscles, fixing your eyes towards that point, all of that is important with respect to this actual tectospinal tract. Now, what's the more important thing for this guy, though? Some of the fibers can go down to form descending motor tracts. Most of them, though, will go upwards as a part of this whole structure here, and they call this structure the brachium of the inferior colliculus. So most of the fibers will go upwards to a special nucleus around the thalamus, and these are called the brachium of the inferior colliculus. Okay, so it forms this thing called the brachium of the inferior colliculus. So this is the brachium of inferior colliculus. All right. Now, once these fibers terminate on these nuclei, what are these nuclei? What are these baby blue nuclei here called? These baby blue nuclei here that are situated on the thalamus are actually called there's a specific body there, okay? Easy way to remember this one is, okay, so it's actually kind of weird. I like to do things a little bit differently, but your eyes, right, they're medial, okay? And your ears are lateral to your eyes. So now, with respect to that, think opposite for the nucleus. So my ears are lateral to my eyes. So the nucleus right there has to be the medial geniculate nucleus. Okay, so they call this structure here the medial geniculate nucleus of the thalamus. Okay, so here's your thalamus. There's a nuclei situated on it called the medial geniculate nucleus. From here, there's radiations that'll be sent out from this actual thalamus. Okay, so now from here, you're going to have these actual auditory radiations that'll be sent to a specific part. It'll get sent to a specific part of the actual temporal lobe. So you know within the brain, here, let me actually draw a small little diagram over here. Let me do this right here. Let's say, here's the brain here, right? So now, you know here's your temporal lobe, right? Here's your temporal lobe. Right on the temporal lobe is an important gyrus, okay? Right here is this gyrus. This gyrus here is called the superior temporal gyrus. Okay, another name for it is they actually call this one the transverse gyrus of Heschel. All right, so they call it the transverse gyrus of Heschel or the superior temporal gyrus. Now, this area is important, okay? It's believed to be what's called the primary auditory cortex. So they believe this area to be associated with the primary auditory cortex. So it makes you aware of the actual sounds, helps you to be able to perceive the sounds or that sound stimulus. There's other areas around it, which are like the auditory association area, which is important for being able to determine the meaning of the sound, right? You know, there's another structure, really important, a little bit posterior to it. So in order for us to be able to really comprehend the sound, there's an area right here posterior to it. You know this area right here, they call this Wernicke's area. So they call this Wernicke's area. So this area plays a role within the comprehension of speech. So again, what does this play a role within? It plays a role within the comprehension of speech. You know why that's important? If someone's talking to you, you're hearing them. You're becoming aware of that sound. That's the primary job of the primary auditory cortex, right? So you're becoming aware of the sound. You're perceiving the sound stimulus. You're relating it with different structures, different nuclei within the brain to determine the meaning and the actual the point of the sound stimulus. Then the Wernicke's area is actually going to get some of those fibers, some of that auditory stimulus to help us to comprehend the speech. Then what do you want to do? You don't want to just sit there not responding to the person. You want to be able to speak to them. So you know what's really cool? The Wernicke's area talks to a specific area here located within the frontal lobe. Let's assume, obviously, this is the right part, but assume that this is the left side, because the Broca's area is only on the left side. So right here in the frontal lobe, there's another nucleus here. And this is called the Broca's area. He's the one that controls the muscles of speech, okay? So he controls, specifically, the muscles of speech. So what happens is, you get the sound stimulus, goes to the primary auditory cortex, you perceive the stimulus. You also have the auditory association area to help us to associate that sound stimulus with previous memories and help us to understand the importance of it. Then it can go to the Wernicke's area so that we can comprehend the speech. But then what if you want to talk back to the person? Let them know that you understand it. There's a fasciculus that connects the Wernicke's area to the Broca's area. They call this fasciculus the arcuate fasciculus. It's called the arcuate fasciculus. It connects to the Broca's area. Now, what does the Broca's area got to do? The Broca's area has to tell specific motor parts. So you know there's motor cortexes. Let's just assume that it's right here. Okay, you have the primary motor cortex and you have the premotor cortex. Let's just assume it's right there. It's going to tell that primary motor cortex, the premotor cortex, even the somatosensory cortex about that. And then what is that going to do? That's going to send down these descending fibers. Where will these descending fibers go to? They'll go to the muscles that control speech so that you can talk back to the person. Let them know that you comprehend the speech, you understand what they're saying. That's really important. So, with that being said, what is this gyrus right here called? Again, just to make it clear and simple, this is the primary auditory cortex, which is within the temporal lobe. And this is where this part right here is called the transverse gyrus of Heschel, or the superior temporal gyrus. So it's really important that we understand that so that we actually comprehend the actual speech. Okay? Because then there's a lot of other structures that will be associated with this. Now, one more thing that's also important is that the inferior colliculus, we didn't say this, but you know the inferior colliculus? It's also communicating with one another. So there's crosstalk between the inferior colliculus, there's crosstalk between the nucleus of the lateral lemniscus as well. So there's so much crosstalk between these actual tracks, just helping to influence the fact that the actual sound or hearing is bilateral, okay, making sure that we can hear from both ways. Now, here's the important part. I wanted to mention something about this nucleus here, the superior olivary nucleus. It's super, super important, really, really important. So we got to mention a couple things about this guy here. So the superior olivary nucleus, I want you to remember three basic functions of it, okay? Three basic functions. It's actually split into two parts, a medial and a lateral. We'll do it real quick. So we have a medial superior olivary nuclear complex, right? And then we have another one, which is the lateral superior olivary nuclear complex, right? The lateral one is specifically important with being able to play a role in the intensity of the sound, okay? So he plays a role in being able to determine the relative intensity of the sound. And I'm going to explain what this means in a second of the sound stimulus, right? So he can determine the relative intensity of the sound stimulus. The medial superior olivary nucleus, here I'm not going to be able to fit it here, let me put it below. The medial superior olivary nucleus, he plays a role in determining the relative timing of the sound. So he helps to determine the relative timing of the sound stimulus. Okay. So now let me explain this because there's one more function that I want to mention for this guy. So again, you have the lateral superior olivary nucleus and the medial superior olivary nucleus. Lateral plays a role within the intensity of the sound. If you had to remember at least one word from these guys, at least remember intensity. And the medial superior olivary nucleus plays a role within the relative timing of the sound stimulus. Now, how could I explain this? Okay. So the superior olivary nucleus, let's say that I'm talking to you, right? And I'm talking straight in front of you. Or I could be talking behind you. Or for whatever weird reason, I'm above you. And I'm talking above you. If I'm talking in front of you, behind you, or directly above you, the sound waves will get funneled into both ears at approximately the same time. So they'll reach the superior olivary nucleus of both sides at about the same time. But then let's pretend you're standing right here, okay? And I'm talking into your right ear. The sound stimulus will reach the right superior olivary nucleus first. And then it'll take a little bit of time, but then it'll reach the left side, the left ear, then the left superior olivary nucleus. So he plays a role being able to determine where the actual sound is coming from. He helps us to play a role, big point here, just the big overall picture. If you don't even remember this, at least remember this point. He plays a role in being able to play a role, he plays a role in localization of the sound, okay? So he helps us to localize the sound, where the sound came from. Is it behind us, in front of us, side of us? Where is it? So he is really important for that. One more thing he's important for. If we talked about this when we talked about the cochlea, but if there's really loud sounds, really loud sounds that could be possibly damaging to your actual hair cells, this guy, he's pretty nice to us. And what he does is, he has some fibers, and these fibers, he activates this bundle. There's a bundle. It's called the Olivocochlear Bundle. So from this superior olivary nucleus, he helps to form this structure here called the Olivocochlear Bundle. And what this Olivocochlear Bundle is it's actually going to be cholinergic neurons, which means it releases acetylcholine. These neurons are going to come over here to the cochlea. So now let's go over here to this cochlea for a second. And now what I'm going to do is I'm going to have these fibers innervating these structures. Particularly though, more of the fibers are going to go to these hair cells. If you remember, what did we say these hair cells were? We called these hair cells, since they were outer, we called them the outer hair cells. So primarily, most of these efferent neurons are going to be going to the outer hair cells. If you remember why, that's important. Remember there was those molecules called Preston? It releases acetylcholine, which hyperpolarizes the outer hair cells. The Preston molecules will no longer do what? They're no longer going to contract. If the Preston molecules don't contract, the hair cell doesn't shorten anymore, it actually elongates. What does that do to the basilar membrane? It bows the basilar membrane downward. If it bows the basilar membrane downward, is that going to cause a lot of shearing of the actual hair cells, the stereocilia? No. If the stereocilia aren't beating towards the kinocilium, is there going to be a lot of activation? No. And so what is that going to do? The action potentials that are being sent down this neuron, it's going to decrease it. So it's a way of being able to protect our hair cells. There's another way though too, one more area. This guy is always pretty important because you know whenever there's a really loud sound, you're becoming very alert of it. There could be a lot of sounds going on in the room, but if there's someone that just speaks out really loud and just screams something for whatever reason, there's a special nucleus, a special sheet of gray matter, which is kind of located around all the way from parts of the midbrain, the pons, and the medulla. This guy is called the Reticular Formation. So whenever there's really loud sounds, high amplitude sounds, the reticular formation will alert our central nervous system, our entire cerebral cortex, to make us alert, aware of the sound stimulus. Some of the information can even go to parts of the cerebellum too. Okay, so now this reticular formation, he's not only going to be important for being able to aware us of the actual sound stimulus, that loud high amplitude stimulus. He's also going to activate two different nuclei, okay? One right here, and then one right around here, okay? So what are these nuclei? Let's say that I have this nucleus right here, and this one is really important. He's going to bring these impulses to a specific part of the actual middle ear. You know there's a muscle right around this area? Actually, hold on, we'll talk about that in just 1 second. Let me bring the other one over here too. There's another muscle, another nerve that's also going to be stimulated. This red marker sucks. Let me get another one. All right, they got this guy right here. You're going to activate another nucleus, which can also move out here and innervate another muscle. So there's two muscles that we're going to innervate. This blue neuron, we're going to call this guy a specific type of cranial nerve. This is actually cranial nerve 5, the trigeminal nerve, okay? We have another nerve which is running right next to it here, to this area, to the middle ear. And this structure is going to be cranial nerve 7, the facial nerve. these are going to go to special muscles in this area. So now, this cranial nerve 5, it's the trigeminal nerve, right? He's going to go to a special muscle. This special muscle that he's going to go to around the middle ear is going to be called the Tensor Tympani. All right, now, let me explain how this works. Let me say here, I have malleus. All right, so here's malleus. Okay. Now, malleus is actually connected to the tympanic membrane, right? So he's connected to the tympanic membrane. So let's say here we have the tympanic membrane like this, right? Well, there's the little part here, the medial part of the malleus. What happens is there's a muscle called the Tensor Tympani. And this muscle, which we're going to draw in blue here to color coordinate, is going to be called the Tensor Tympani. Okay, it's right around where the eustachian tube is, where he actually is going to be connecting. Now, what he does is when the actual cranial nerve 5 comes over here and innervates it, stimulates this muscle, he's going to contract. And when he contracts, he pulls on the malleus. When he pulls on the malleus, guess what he does to the tympanic membrane? Tightens the tympanic membrane. Thus the name Tensor Tympani. It tightens or tenses the tympanic membrane. Now the tympanic membrane isn't going to be as sensitive to that sound waves. So it helps to dampen the actual sound waves. So it helps to dampen the sound stimulus. And that is just so darn cool. So another way of our body being able to protect us from certain situations which could damage our hair cells, okay, is by activating this muscle right here. And again, what is this muscle here called? This muscle is called the Tensor Tympani. Now, what about this other one? What about this other muscle? The other muscle is going to go to, from this actual facial nerve, is called the, this marker's good, stapedius. So it's going to go to a muscle called the stapedius. Now, the stapedius is a pretty cool muscle, and what it does is it actually connects to, yeah, you guessed it, the actual stapes, okay? Now, what happens is stapes is responsible for being able to tap onto the oval window, creating fluid-filled vibrations. And whenever there's loud sounds, it's going to tap really, really hard and really fast. What we can do is we can have this stapedius muscle become stimulated by the facial nerve. And what's it going to do? It's going to pull on the stapedius. And it's going to prevent the stapedius from tapping on the oval window. If it pulls the stapedius, the actual stapes won't tap on the oval window as much and as hard. It's not going to create as much fluid-filled vibrations, which is going to prevent less hair cells from being activated. Again, another way of being able to dampen the sound stimulus to prevent damage to our inner hair cells. So I think that's a beautiful, beautiful thing. Okay, the last thing I want to talk about, because it's relative. It happens in some people, and it's an unfortunate thing. There's a pathology called an acoustic schwannoma. They call it acoustic schwannoma. Now, this is actually, it's not too common. It can happen, though, and usually the only way to be able to see this is sometimes you'll see the person can exhibit like vertigo. They can have tinnitus, which is just basically like the ringing of the ears. They might even have other types of problems where they actually might even have a loss of hearing. Okay, so an acoustic schwannoma. What's actually the cause of this? What's the cause of an acoustic schwannoma? Okay, so let's actually say here, very, very briefly, let's say we have a cell, all right? And the cell, your cell has specific types of, genes, right, that's occupied within the nucleus, right? Here's your DNA. There's specific genes that are regulating specific types of protein synthesis. Let's say here's this gene right here. This gene right here is called neurofibromatosis type 2 gene, right? What it does is it produces a special type of protein. And this protein that we're making actually gets secreted out of the cell, right? It gets secreted. But what it does is it binds onto an actual protein here on the cell membrane. There's a protein here in the cell membrane. This protein, it's called CD44, right? What happens is this neurofibromatosis type 2 gene produces a protein called myelin. And this myelin binds onto the CD44 protein. Okay, what is this protein here called? It's called myelin. This myelin protein binds onto the CD44. And what that does is it allows for this myelin protein, the CD44, and you know CD44 is actually connected to some actin molecules inside of the cell. What this does is, once you have this whole complex here, so for example, let's say that here's the myelin protein, and then right connected to it is actually going to be CD44 protein, which is a part of the cell membrane, and then connected to that is going to be the actin molecules, right? And the actin molecules are helping it to be able to support the cytoskeleton. So now here's the cell membrane, this part that I'm drawing here. What's important is that this myelin in CD44 is actually connecting to the extracellular matrix. Why is that important? In a situation in which someone has an acoustic schwannoma, guess what they don't make? They don't make actually this myelin protein because there's some type of damage to the neurofibromatosis type 2 gene. If they don't make the myelin protein, it can't connect to CD44. If CD44 and myelin are not connected, that means that the cell isn't actually connected to the extracellular matrix. What can it do then? It can start dividing. And as it starts dividing and dividing and dividing, guess what it starts making? It starts making a huge mass. And usually this mass occurs in like around the internal acoustic meatus and actually kind of around the cerebellopontine angle. And what it does is it actually can compress the nerve fibers. And obviously this can lead to hearing loss. It can lead to tinnitus, the ringing of the ears. Depending upon how big it is, it can actually affect our balance too, because it might affect our vestibular system. Okay, and this last situation here is, you know, these actual little ossicles, these little bones here, sometimes due to frequent infection or as you just get older, it starts actually developing a lot of fibrosis. And it causes these actual ossicles to become thickened and hard and not able to move as much during the response to the sound waves. And that can actually cause a specific type of deafness. So what is these actual guys here? This actually happens due to otosclerosis. Otosclerosis is actually going to produce deafness. It can produce deafness, but this is called conductive deafness. Okay? Other types of conductive deafness is if you have earwax that's really, really building up within the external acoustic meatus. That can also affect the actual sound waves. But the acoustic schwannoma, this is actually due to effect of the actual inner ear structure. So this is actually called a sensory neural deafness. Okay. All right, engineers, so in this video we talked about the auditory pathway, specifically following the cochlear branch of the vestibulocochlear nerve. Guys, I really hope it made sense. I hope you guys did enjoy it. If you guys did learn a lot, if it did make sense, please hit that like button, comment down in the comment section, and please subscribe, guys. All right, also, if you guys want, please go check out our Facebook, our Instagram, and even our Patreon. If you guys have the opportunity to donate, we would so appreciate it. All right, engineers, as always, until next time.